Okay, hello, welcome everybody, and thank you so much for coming to our webinar today. I'm going to share some slides with you. My name is Carol Merchart, and I am the Executive Director of Access. This webinar series is presented by Claris Therapeutics, and we thank them for their support. The mission of Access is to help individuals with one or more extra X or Y chromosomes and their families to lead fuller, more productive lives. We do this through support, education, research, and helping find treatments. I hope everyone is aware that Access has a helpline. We have a brand new number. It's listed here, 267-338-4262, or you can email us, helpline@genetic.org. Trained volunteers will help answer all of your questions, and we try to connect people with a peer, so someone who has a prenatal diagnosis can talk to someone else with a prenatal diagnosis or someone with that same condition. We have support groups on Facebook. They're grouped by condition, and they're also grouped by geography. Hopefully soon we'll get back to our in-person support groups. They're spread out throughout the United States, and we're really just happy to be able to bring people together. We're really thrilled that because of a grant from the Kozlowski Family Foundation, we now have a continuing medical education course on Klinefelter syndrome in adults. Please direct your doctor to go to genetic.org/CME, and they can earn free CME credits. We're hoping that this is the first course of many we can do in the future to help educate medical professionals. We have a library of scientific papers, such as the one we're going to talk about today, and you can find that on our website. We also want to make sure you're aware of the Access Clinical and Research Consortium. We have a group of clinics throughout the US, and we also now have several in other countries, including Canada, the UK, and Denmark. Visit genetic.org/clinics to find a clinic near you and to see who's part of our clinical consortium. One of the great things about this clinical consortium is actually bringing us to what is happening today, which is that Dr. Nicole Tartaglia, who I know you all know, from Denver, she put together a journal, a special issue of a journal with many papers on the X and Y variations, and we've been presenting many of those to you. This webinar and several other webinars will be ending up on our Access YouTube channel. The channel also has over 100 recordings along with these webinars that are from past conferences on most of the topics we'd be interested in, from fertility, disclosing the diagnosis, mental health, research paper, legal issues. So please make use of that great YouTube channel. I warmly invite you to join us at our first ever Access Virtual Family Conference. We're going to be online this year. We hold a conference every other year, and we're really sorry that we can't be in person this year, but we're just going to hope to make it a great experience for you. It's going to be over the last week in June. You can find the information on our website, and you can purchase your tickets right now. What we like to show, and this is a great example of what we're doing today, is Access community and how we work with researchers. So all of our community members may go to a physician researcher, participate in a study, whether that's at NIH, it might be at one of our clinics. Then those clinical studies, we help the scientists and researchers recruit individuals for those clinical studies. They present them at scientific conferences, but many, like Dr. Mountford today, is presenting it to us on this webinar. So it gets back to the Access community. So today's talk is do secondary copy number variants explain differences in severity in people with sex chromosome trisomy. And we are really thrilled to have Dr. Hayley Mountford with us here today. And I also want to let you know that it's complicated scientific information, but Zoom just added a live transcript button at the bottom, and you'll see a thing that says CC in it. So if you wanted to make a closed caption so that the words are coming up as we're speaking, please feel free to try out that feature. And I'm very happy to present Dr. Mountford. Thank you.
I'll just share my screen. We practiced this. Okay, can you see that okay? Yep, thumbs up from Rick there. Great, so it's really exciting to be here today, especially because we've all been in lockdown for almost a year. So it's our lockdown anniversary here. And frankly, we don't get out much at the moment. So being able to do this webinar for a community of people is exciting, and I'm looking forward to all your questions at the end. So today I'm going to talk to you about secondary copy number variants in trisomies and what they might mean for severity that you see within the trisomy spectrum. So first of all, a bit about who we are. So this project was a collaboration between Oxford University and Oxford Brookes University. And the main driver of this project is Professor Dorothy Bishop, and she is primarily a psychologist. She also does a lot of research into replication studies, so taking studies that exist in the universe and testing them in a different way to make sure that they replicate and that they're statistically robust. And then also Dr. Diane Newbury, so she's my PI, so she's the head of my group, and she's a geneticist who generally focuses on language disorders and the genetics that underlie them. So these two are language powerhouses, and this is really the sort of aspect we're mainly thinking about in trisomies. So the work was funded by the Wellcome Trust Programme Grant and also the European Research Council. And we also had ethics from Berkshire NHS Hospital. So I probably don't need to explain any of this to this particular audience, but I'm going to just cover it anyway. So this is a chromosomal karyotype. So most people typically have 22 pairs of autosomes. So these are numbered 1 through 22, and they're in size order. So 1 is the biggest and 22 is the smallest. And then we typically have a pair of sex chromosomes. So these would be XX in a female or XY in a male. And the karyotype that you see here is referred to as 46XX or 46XY. So this is what you'd find typically. And this little picture here, I found this is the best picture of some actual chromosomes. So this is what they actually look like. They, the X is enormous in comparison with the Y. The Y is a tiny little thing. So now we move on to sex chromosome trisomies. So sometimes there is different terminology around these. So they can be called sex chromosome aneuploidies, but the term aneuploidy can also mean that one is missing. So in this particular talk, I'm going to use the term trisomy because we're only talking about when there's an extra chromosome specifically. We're not doing anything on anybody with a missing one or more than one extra. So sex chromosome trisomy refers to having 3 copies of the sex chromosomes. And we are looking at 3 different subtypes. So there's 47XXX, which is 3 copies of the X chromosome. And that is sometimes called trisomy X and occurs in around 1 in 1,000 females. Then there's Klinefelter syndrome, which is 47XXY, and this is somewhere between 1 in 500 to 1 in 500 to 1 in 1,000. And finally, Jacob syndrome, which is 47XYY, so 2 extra Y, an extra Y, and this is about 1 in 1,000. Now, these estimates I got them from the Access website and a couple of really good rarediseases.org and rarechromo.org, but they're generally really thought to be an underestimate because there's a lot of people who potentially have an extra sex chromosome who don't ever know and don't ever find out. So the population estimates they're probably more frequent than these estimates indicate. So as you're probably aware, there's a huge variation in the symptoms and the presentations that you might find in somebody with a sex chromosome trisomy. So the ones that we're particularly interested in are the neurodevelopmental differences. So these are things like intellectual disability, behavioural issues, and autism spectrum disorder. There can also be attention difficulties, or they can have dyspraxia, which is a motor condition of coordination. And particularly interesting to us is that often people can have a difficulty with language and difficulty with speech. So this is called heterogeneity. So this is when there can be a huge range of different symptoms, even with the same apparent underlying genetic cause. And as well as having a different selection of these symptoms, they can also range in severity. So one person may have extremely mild language disorder, one person may have a very severe language disorder. Somebody else with the same trisomy may have no issues at all that are obvious. So our focus is on language, and in this little graph at the bottom, this is just to show you the broad differences in people with trisomies compared with controls. So this is called a pirate plot. I don't know why, but I think they look like slugs. I call them slug plots, so that's what I'm going to call them. So the blue slug is the trisomy group. So I use SCT as the abbreviation for that. And then that's compared to the controls. So the scale, a little bit counterintuitively, is if you're really good at language, you score 30. If you really struggle with language, it's minus 30. So the trisomy group finds language substantially harder than the control group. So the thick black line across the middle, that's the average, that's the mean of the two groups. But within each group, you can see there's a huge sluggy spread all the way down. So there's an enormous range of abilities within that. So some people are basically fine, and some people have a very profound difficulty. So next, I'm just going to quickly explain what copy number variants are. So they are very, very similar to what you probably know about trisomies, but it refers to a much smaller chunk of DNA. So these copy number variants can be on the autosomal chromosomes, which is the 1 to 22, or they can be on the sex chromosomes. So usually they appear a bit on the X, but not very much on Y. But I think that is because it's very small and not that much is looked at on the Y. You never really hear about it that much, sorry. So essentially, copy number variants are chunks of DNA that are either missing, which is a deletion, or an extra bit, which is a duplication. And they're very similar to trisomies or aneuploidy, but it's a much, much smaller region. It can also include some which you may have heard of, which have got a whole arm of a chromosome, which might be missing or extra. And when it's a large one like this, you can actually see it on the karyotype. So you can see it down a microscope. If you look at the chromosomes, you can actually see a difference. So in my little, can you see my pointer? Yeah, okay. I'll stop waggling it around. So this is a wild type chromosome here. So this is actually the X again that I've reused throughout this talk. And then if you imagine this pink square here is the CNV region, which has been deleted. So then the chromosome shunts up and it's just missing this bit and you get a smaller version. But then you can have the same thing if that is duplicated. It doubles up. So therefore this chromosome is much larger. It's a much larger version and it's got 2 copies of this bit here. Or, which is more common, they can be very, very small. So they can be a microscopic, beyond microscopic region of the DNA. So it can be so small, it's even a single gene or maybe even part of a single gene. So these are not visible through the microscope. These have to be detected in a really different way. So an example of this is a deletion syndrome called 15q11.2. And people with this deletion sometimes have neurological difficulties, developmental delay, language difficulties. They can have motor delay, ADHD, or autism spectrum disorder, or none of those things. So again, there's a really varied presentation. Some of them can be very profoundly affected, or again, can be a carrier and not have any clinical presentation at all. So this is an example that in neurodevelopmental disorders and in CNV disorders, heterogeneity is really common. It is the rule rather than the exception. So we see this all the time, that if you have this particular thing, you might have it, you might have a mild version, or you might have none at all. So it makes them very hard to study. So back to copy number variations. So everybody has these in their DNA. So usually people have about 4 or 5 that you consider important ones, but it's not very well understood what they actually do. They vary in size. So some are really big, some are really small, and they vary in where they are. So they could be anywhere in your genome. And everybody has a slightly different combination. So some of them are very, very common and lots of people carry them, but others are really rare. So it might only be in one person or just in your family. And they do, and everywhere in between as well. So some of them are sort of in the middle. Most of them are benign. So this means that they don't really do anything. They don't cause us any problems. There's nothing detectable or damaging about them. And they just persist in our genomes. And we don't really understand why that is or what their purpose is. Some of the others on the other end of the spectrum are predicted to be damaging. So this means that they can be detrimental to the cell and to your body, and then you may have a disease or disorder as a result of a damaging CNV. So because of this, this variation and how common they are and how rare they are, and that they're always different, it's very, very hard to predict which ones are going to be damaging, which ones are going to be benign. Also, to add to the complexity, some of them are inherited, so they might be shared by several family members, or sometimes they just appear in somebody's DNA, and then this is called de novo. And because this is a sex chromosome trisomy group, you'd probably be very, very familiar with the idea that sometimes things can just appear in a person and nobody else has that condition. So CNVs have got a role in language disorders. So we know that they play a role in neurodevelopmental disorders more broadly. Usually these are very rare CNVs and they are very predicted to be damaging. So these are some really bad ones that you probably don't want. And we think that the difference with some of these is that they cover genes that are critical for brain function. So that they're covering, that the CNV has deleted or duplicated genes which have an important role in our brains as they develop. And if we're missing a copy or maybe missing 2 copies, we actually can't function and develop as we need to. So another idea of this is, particularly in language disorders, is that sometimes people with these disorders have more copy number variants. So they're not necessarily damaging ones, they just maybe have a little bit extra. And we think that maybe this makes them more susceptible to developing a language disorder or another neurodevelopmental disorder. So we know, my supervisor, Diane Newbury, published a paper back in 2015 where they found that individuals with language disorders on average had at least 1 more copy number variant than in controls. So it's a very, very subtle change. It's very subtle stuff. It's not a huge difference. And it's very hard to interpret what it actually means. But there's also a similar story in other disorders. So in autism, in dyspraxia, and intellectual disability, where you see this slight increase in people who've got these disorders over controls. It's very, very subtle. Just keep that in mind as we carry on. But bringing all this together, this is a very strong case for looking at the role of CNVs in neurodevelopment as a whole. So now back to sex chromosome trisomies. So as I explained we think that CNVs play a role in neurodevelopmental disorders. So this is from maybe having slightly more, or also maybe having a very rare damaging one. So we know that there's a role in neurodevelopment. And a couple of years ago, 2 really important papers came out on sex chromosome trisomies and copy number variations. And so we then thought, this is a good thing to look into, because this might actually help to explain some of the variation that you see in phenotypes. So the first paper was by Rocker and colleagues in 2016. And what they did was they looked specifically at Klinefelter syndrome. So they had 94 people with Klinefelter syndrome, and they had 85 controls. Oh, look, I wrote 42 and 43. I didn't mean that. I meant 46. And so what they did is they took on all of their X chromosomes, they did a really deep analysis to look for copy number variants, specifically on the X chromosome. And they found that in the people with Klinefelter syndrome, they had on average 4.5 copy number changes just on the X chromosome, whereas the people in the typical XX and XY groups had about 1, just over 1 each. So this is a huge difference. They have people with Klinefelters, they found had 3 times more CNVs on the X chromosome. This is a massive difference. So we wanted to look into this to see if we could find the same and if it replicated. Then the second paper was by Legall and colleagues in 2017, and they did a slightly different study. So this time they looked at 3 different subgroups. So they had 14 patients in total, 4 XXY, 4 XXX, and 6 XYY. And they looked at copy number variants across the whole genome this time. So they looked at everything. And in 7 of these patients, they found pathogenic CNVs. So what I mean by this is they found copy number changes that have been previously reported in other patients. They're a clinical diagnosis of, if you have this copy number change, you probably have this disorder. So they're very strongly linked. They are, it's diagnostic. In another 2, they found copy number changes that they felt had less evidence, but they were still very strong. So likely pathogenic is still a very, very strong, we think you have this disorder because we found this copy number change. And in the final 5, they found potential ones. So ones that fit the bill, but weren't quite as didn't quite have the clinical evidence. So again, this is huge. In their 14 patients, they've got a 50% rate of identifying secondary CNVs that they think are contributing to these patients' phenotypes. So again, we thought, wow, this is huge. This explains it. So we took these 2 very strong studies and tried to replicate to see if we could find the same thing in our study. So from these papers and the things I've mentioned about neurodevelopmental disorders in general, we put together some hypotheses. So we took the evidence from all of that and we came up with 2 ideas. So the first one is burden, and that is, do people with sex chromosome trisomies carry more copy number variants on average than controls? And if they have a more severe disorder, are they then more likely to have these secondary CNVs? The second theory, we call this the double hit theory, where we think perhaps there's the secondary CNV is adding to the trisomy to give a more severe phenotype. So if you have the CNV by itself, you have a middle phenotype. If you have a trisomy by itself, maybe a middle phenotype. And if you have 2 together, then you have a much worse phenotype. So what we did was we designed an experiment. So a lot of this is about data and replication, because it's really important in research to layer evidence. And if you want to take it into a clinical setting where it actually has a direct impact on patients, it's really, really important to validate independent studies and to really, really show in multiple layers that it really is real. So what we did, so this all happened way before I came along. So there was an enormous effort, and they recruited 125 people with sex chromosome trisomies. Some of you might be here, or maybe members of your family. That's really exciting. Say hi if you were. So they were all children aged between 5 and 16 years old. And all of these people knew about their diagnosis. That was really important ethically coming into the study. And they were recruited partially from a previous study that Dorothy did, but also through various groups, support groups, and also through the clinical genetics services in the UK, and also self-referral through Facebook. So it's a good way of kind of expanding and recruiting people. And so in the end, there were 40 people with XXX, 42 with XXY, and 43 with XYY. So they did very well in having an even spread of people with different subtypes. So something to bear in mind with this is that how the people in the studies were first found out that they had a trisomy. So essentially, some people in the study were referred because they had neurodevelopmental difficulties. So they had problems with language or coordination, and they were having investigations because they were struggling. And we call them the high bias group. And then the other group is the low bias, and they were the people who they got their diagnosis from screening or from medical investigation of something else. So they tended to be people who didn't have such overt neurodevelopmental difficulties. So it's just important to think about that when we're talking about looking at this population as representative of everybody, because it really isn't. It's a very selected, skewed population. And then we had a comparison group. So they were selected because they'd all had the same testing, which we'll get to in a minute, and were already recruited by Dorothy and her group. So they were actually twins, twin pairs, and they were all recruited into the study because it was on language difficulties. So again, because we're looking at neurodevelopmental measures, a lot of these children already struggled with language. So again, they're not representative of the typical population. They're a slightly skewed population too. But that makes them an excellent control group, because if we find anything, then it'll be even stronger signal than it would have been with a typical control group. So we took one of each pair. So because if you have siblings, that gives you sort of a false signal, because we're looking at genetics. So we took 181 individual twins into the study. And also, just to link in with what I said earlier, that we found previously that children with language disorders were more likely to have an extra copy number variant. So that's sort of carries through into this, that we already know that it might be enriched in that group. So it's not an ideal control, but they were the group that we had available to us at the time. So we had 82 girls and 99 boys. So all 300 of these children went through the same testing battery. So this was an epic effort, which was mainly done by 8 research assistants who did an enormous amount of work to put this together. So there were a whole bunch of different tests that were done. Some of them were done over the phone or by parental questionnaires. And then a load of stuff was done actually in person. So they had to go visit all of these children in their homes, or the children had to come to the centre. And they all did all of these different tests, and also took a saliva sample to get a DNA sample. So first of all, I'm going to tell you about our main measure of interest. So we took all of those different tests and took aspects of them to add up to give a global neurodevelopmental index. So what this does is it takes a bunch of information and consolidates it into a scale of 0 to 6, where 0 is no problems and 6 is lots of problems. So to score a point on this scale, you have to have a fairly severe disorder. So this doesn't reflect children with a very mild or moderate disorder. This is quite severe. So this is having a formal diagnosis of something, having special support for it in school, or having serious conduct issues. So to score a point on this scale, it has to be a fairly severe phenotype. So the maximum was 6. It is possible to score more than 6, but it was a scale of 0 to 6. So this here's my slug plot of this. So the trisomy group are in the blue slug, and the comparison group are the pink slug. And 0 at the top is no problems, and 6 is lots of difficulties. So you can see that the trisomy group generally on average had a lot more difficulties than the comparison group. So again, the thick black line is the average. So there's quite a big difference between the two groups. So then we looked at language factor. So this is, again, taking a bunch of different tests and consolidating them into one number. So this reflects all aspects of language ability, vocab, being able to get your words out, being able to do things in the right order. So it really, and being able to understand instructions. So it really took all of language and we stuck it into one measure. So again, the slugs show that the trisomy group had poorer language skills on average than the comparison group. So again, it's also the top, so a big number, so 30 shows good skills and minus 30 shows severe difficulties. So then finally, this is performance IQ. So this is a measure of non-verbal intelligence. So this is a way of measuring IQ and it allows for children who do struggle with language. It's a way of measuring IQ even though they might not be able to speak very well. It looks at a different aspect of IQ. And I didn't have a slug plot, so I got a dog with a scarf on. So back to CNVs. So we find CNVs by using a technology called a genotyping array. So what this does is it looks at a whole bunch of different changes across the whole genome all at the same time. And these are individual base pair changes. So this is actually this slide here is one genotyping array. And each of these little squares is one individual sample. So this would be one person's DNA. So our genomes contain about 3 billion of these bases. So this is the A, the G, the T, and the C. And these letters combine together to make a code, which then is organized into genes. And then these genes are translated into make proteins. And then these proteins build our cells and control our bodies and build our fingernails and things like that. So this genetic code is packaged into chromosomes, which I'm sure you're all really all across the chromosomes. And it basically is like a blueprint. It contains the instructions for making a body. So sometimes these bases, these A, G's, T's, and C's, can have a spelling mistake in them. And these are called, we call these variants. We sometimes call them SNPs, but that stands for single nucleotide polymorphism. So it's just jargony. So I'll try and call them variants. But you can sort of think of them as individual spelling mistakes. And it's the same with the copy number changes. Some of them are harmless, they don't do anything. Some of them are very bad, and if you have them, you probably have this disorder or this disease. So I've just got a little picture here to show you. It may have been reused from earlier. So if you imagine on your chromosome, we have 2 copies of each of our chromosomes. And if we have, imagine that the pink box is the base letter A. So this person has got 2 A's, 1 in each copy at this site. And this is called wild type because it's almost like the default setting. So this is what is most commonly found at this site. And it's called homozygous because they have one version of it. So then the next one is this person has slightly different. They have an A and they have a G at the same site. So this is called heterozygous. So this is when you have one copy of the wild type, which is A, and then one copy of a different one, which is G. And then this person has got 2 A's. So they are homozygous again. So they've got one type for the alternative allele. So then these SNP arrays and these genotyping arrays that I mentioned, look at these individually, but on a huge scale. So it simultaneously looks at about 4 million of these variants all at the same time. So mainly these are, they're chosen because they're different in the population, that we all have different combinations of them, they're quite common ones. And so these span chromosomes 1 to 22 and the X chromosome. Poor little Y doesn't really have many. So they don't tend to look at that one. I told you it's neglected. And so when you've looked at this across lots and lots and lots and lots of SNPs of variants, this is what it looks like. So in the middle, you have the A's and the G's. So this is the heterozygous. Then all of the wild type ones get shoved to this side and all the A's get shoved to this side. So this is what you'd look like if you have 2 copies of a chromosome in your genome. So then it gets a bit more tricky when you look across different chromosomes, because in this one here, you can actually see a deletion. So it comes up as in the middle, there's none of the heterozygous. They only have one homozygous on this side or homozygous on this side. So that means that you can infer that this is a deletion in this region. Then on the other side, we've got a duplication. So they've actually got 2 copies of this, 3 copies of this region. So now you have these double parallel blue lines, which shows that there's an extra copy. So this is what you can also use this to look at trisomies on a genotyping array. And this here, the duplication, is what it looks like when you have 3 copies of X. So you can actually use these to look at trisomies as well, and you can get that information from them. So first up, we looked at the burden. So this is whether people with more severe phenotypes have extra CNVs, or maybe their CNVs cover a larger region. And so this is there's no slugs this time, but it's a bit. So I'll just draw you down attention to the table first. So in our trisomy group, we found there were about 5 CNVs per person. And so the average was, I think, no, I've forgotten, sorry. We had about 5.2 CNVs per person. In the comparison group, there were just over 4. So we are seeing that perhaps people with trisomies may, on average, have slightly more than the control group, but it's a very, very small difference. And then we also looked at the low bias group. So these are the people that they're more representative of a population screen. And we saw very, very similar numbers. So the people in the trisomy group had very, very slightly more on average than the comparison group. And then that data is represented in this graph. There's a lot going on in this. Basically, the red are the trisomy group and the black dots are the comparison. So when you look at this, there's essentially no difference. They're very, very similar. So in some cases, there's hardly any copy number changes, and in some cases, there's lots of copy number changes. But there's not an obvious difference between the two groups. And so I've mentioned a couple of times that one of the key things about the study is that it's focused on replication. So we're trying to make sure that everything is statistically robust, which means that no, it's not. So we can see a very, very subtle difference, but it's not statistically significant. So it means that we really didn't find that. We can't say that we found that there was an increased burden because the difference is so, so subtle. And we did some very whiz-bang statistics on it to try to make sure that what we were seeing was really, really reliable. And as a result of that, we can say that no, we do not think that people with trisomies have more or larger copy number changes. So the next section is, well, what do these copy number variants do? How can you tell whether they're going to be bad or whether they're just natural variation? So we thought perhaps there were some of the people with severe disorders, perhaps they had more pathogenic copy number changes than the people who had mild or no neurodevelopmental disorders. So with benign CNVs, so we tend to think of these as very harmless variation. They're often more common in the population because they're not really doing much. So they don't have any effect on the people carrying them. And we usually assume that they cover genes that are, the genes that they cover, that they've deleted or duplicated, are very, very tolerant to changes, that the genes can still function, they don't mind. It's business as usual. Whereas with the pathogenic ones, we think the opposite. So we think that pathogenic ones are very likely to cause disease or disorder in some or perhaps all patients and carriers, that because of this, that they must be covering genes that are critical to function, that we absolutely need 2 copies of, and we need them to be working. And as a result of that, these are usually very rare. So they, the fact that they are rare shows that they sort of support that they might be quite damaging to carry. And we find out about this information by using a whole load of publicly available data sets. So these are absolutely key to this type of work. Without them, we would not be able to do it. So we used a database called ClinVar, which is where clinicians report when they've had patients where they found a pathogenic CNV, they record it, they record the phenotype of the patient, what some of their neurodevelopmental disorders. We can get a lot of information, and it's particularly important because sometimes there's only 1 or 2 patients in the entire world that have that exact CNV. Then the other database we use is called Nomad. It's not gnomes. This is really cool because it's got all the SNPs and all the genetic variants from 100,000 people across the world. So we can use this data to really accurately tell how common or rare something is in the population. And then that information helps us to know if it's potentially pathogenic or if it's just benign variation that lots of people have. And really, these databases have absolutely revolutionized what we do. We could not do it without them. So we use these to then look at the individual genes that have been deleted or duplicated. And we said, how likely is it that this gene can tolerate being deleted, or is it a gene that absolutely cannot be deleted? It's really, really, really important. And in the Nomad database, they've very kindly done all this for us by using their huge cohort of 100,000 people. So you can tell by how common a change is as to whether a gene is tolerant of losing its function or not. So this is all put together in a single score. We love single scores. It's really, really good. Called a PLI score. And this stands for probability of loss of intolerance score. Again, horrible acronym, horrible. So I'll just call it PLI score. And it basically, if it's 1, it is not tolerant at all. If you lose a copy of that gene or that gene is broken, it will not work. You will be ill. And at the other end of the scale is 0, which is very tolerant, doesn't care. You can delete it, have extra ones. It's very, very tolerant of loss of function. So what we did is we took the PLI score, so how tolerant each gene is, and we added them up for each CNV. So some CNVs have 1 gene, some of them have no genes, some of them have lots of genes. So we added up for each one. And so then this gives us an idea about, for each copy number change, some of them have got a score of a very, very low score. So we think that probably they're not going to be that damaging. Whereas others, the highest score we got was 8. So that particular change covered 8 genes which don't like to be disrupted. So you would think, therefore, that the person who had that deletion would be very ill. But it just didn't work out like that, unfortunately. So if you, so on this graph, we've got down this side, this is the score, so the neurodevelopmental score. So where you've got 6 is no problems in this case, and no, sorry, that's the other way around. 6 is lots of problems, and 0 is no problems at all. Then along this axis, we've got the total PLI number that they scored for all their CNVs. So we have some people in this group who got a huge number of a really high score of 8. So that's 8 different genes that don't like to be changed or affected, but have got very, very hardly any or no neurodevelopmental phenotype at all. So it's pretty clear that there isn't one rule here that fits everybody. It's a very individual thing, and that we can't sort of broadly say that this rule fits everybody. So we also found that overall between the two groups, that on average, the trisomy group had an average PLI score of 0.87, whereas the comparison group had 0.71. So there was, again, absolutely no difference between the two groups. So a little bit more detail about this data. I've sort of already said this, I think, that we can't show that there's a clear link. This does not explain it. So because CNVs with a high PLI score does not equal having neurodevelopmental difficulties. And there is no clear link between gene tolerance prediction and the severity of each person's individual phenotype. So at the individual level, it was a little bit more interesting, but again, it doesn't fit a broad model, and we can't say that this explains it. So now I'm just going to go back to the papers that I told you about in the beginning. So this is the Legall paper. So they looked at 14 different people with a range of trisomies. And so they found, if you remember, 50% of their people with trisomies had a pathogenic copy number variation. So half of their people had an actual clinical change that they detected. Whereas in ours, we only found 9 in 125 people. So we are not even slightly in the ballpark they were. We found that just over 10% of people with a trisomy had a secondary copy number change. So that's a huge difference. That's nowhere near the rate that they found. So by this point, we were thinking, I'm not so sure this explains it. Then the other paper, so the one that only looked at people with Klinefelter syndrome and looked just for copy number changes on the X chromosome. So again, we just couldn't replicate their results. So we only found 5 people with a secondary CNV on the X chromosome, whereas they found an enormous amount. And we just didn't really see a big difference between our trisomies and the controls either. So the controls, just about 4% of people had them, whereas with the trisomy group, it was just about 6%. So not a huge difference. And unfortunately, we couldn't test the X chromosomes for the people who had trisomy X. So this is an artifact of the method that we used, and that we couldn't really tell accurately if they had a copy number change. So the people who did this original paper used a different method that's really, really good at testing specifically X chromosomes. The one that we used wasn't so good, so we couldn't include those people in this particular analysis. So we were sort of left wondering why we couldn't replicate either of these studies. And there are a couple of reasons. So the study that was on the Klinefelter syndrome patients, where they just looked at all CNVs, we used a different technology, and we also, the Klinefelter syndrome was just on the X chromosome. So we used a different technology, as I said earlier, and we had a very, very stringent inclusion criteria. So where we found a CNV, and we called a CNV, we are absolutely confident that it is real, and we deliberately did that because we wanted to be certain of what we were including, rather than be a bit more generous and include some that may not be actually correct. So we didn't find a statistically different difference between these, so we couldn't replicate this. Then with the second study that looked at copy number changes across all of the chromosomes, again, we didn't even get close to their diagnostic rate. Now, we think that this is because in their paper that they only included patients where they suspected that there might be a second copy number change. So this would be patients who have a very profound, difficulties. So at the extreme end of the neurodevelopmental scale. So these children would have been at a 5 or a 6 on our global neurodevelopmental index. So we think that they tested people who were more likely to have a second CNV, whereas we tested the full range of everybody. So then we'll just go back to the hypotheses. So burden, did people with sex chromosome trisomies carry more CNVs in general that we see in language disorders and other neurodevelopmental disorders? Well, there was maybe a very slight signal on average between the two groups. Perhaps people with trisomies carried 1 extra CNV than the control group. Then the second hypothesis, the double hit idea, is there a secondary CNV, and does this result in a more severe phenotype? Very modest enrichment in the trisomy group, but not very big. And no, it's not statistically significant. We cannot say that clearly that we found a significant difference, because we really didn't. Any differences we saw were extremely subtle, The only way that you could get a more robust result would be to have much, much larger studies with hundreds of each individual trisomy subtype, or ideally thousands. You'd have to do an enormous study to really see a result that you could be confident in. And we predicted that 40 would give us a robust result, but the changes and the differences were so subtle that we actually couldn't see a robust result. So it's a very modest change that might be able to be replicated down the track, but not by this study. This study did not find an association. So with these individual differences that we see in trisomies and why some people are unaffected with neurodevelopmental disorders, whereas others are very severely affected, we can't really say that we can explain it. We saw a mild trend, but nothing that we could clearly say this explains the differences. It's a very individual thing, and each person is going to have a different case in many ways, and there's not one size fits all that we can use to explain all of these differences. Some of the children in our experiment did have secondary CNVs, and we think that some of them are likely to be contributing to some of their disorders that we see. But on the other hand, we also saw people who carried secondary CNVs but had no neurodevelopmental difficulties at all. And most people, there weren't any that could actually explain any of their condition. So really, copy number variants are interesting, but the interpretation of them is very, very complex. And this is improving all the time as we get more information from these large databases, but there's a really long way to go. And so anytime a copy number change is found, there's always a lot of caveats and a lot of, well, maybe it could be this or maybe it could be this. There's no clear answers for you at all yet. So my take-home message from this is that copy number variants are important in neurodevelopmental disorders, and they probably are important in trisomies as well. And that in some cases, there probably is an underlying variant which may explain some of the severity of phenotype that you see. But probably for most people, they are not important or not significant. So I would maybe suggest that if you have a child who has a particularly severe phenotype, it may be worth looking for secondary CNVs. I don't know what it's like in the US, but in the UK, usually once you found a trisomy, that's it. No more investigations get done. That's the answer. And it may be possible that there are other things going on which do explain some or part of the phenotype that you see. But this is all very much in a research context. I don't know if that would even be possible. This is all, research rather than clinical. But I hope that one day this will be clinically available. And so with that, I just want to say a couple of thank yous. So mainly to Dorothy and Diane for running this study, and the army of research assistants who did all the hard work in testing so many people. And I'd also like to say thank you to the study participants and their families who put in a lot of hours and came along to the clinic and did all the testing, and we couldn't have done this without you. So thank you very much.
Thank you so much for that excellent presentation, Dr. Mountford. We really appreciate it. So obviously there's been a few questions.
Yeah, I could see the thing popping up.
So I guess one of the questions really is, does this study disprove the first two other studies? Does that mean they were false? Like, what is the take home on this?
They weren't false. Where they found copy number changes in those patients, I believe that that's what they found in that individual patient. But I think that they may have cherry-picked who they tested based on a likelihood or suggestion that there may be another change going on. So it's the actual science of what they did is solid with respect to each individual person. Looking at it in the context of all people with trisomy, it's not that similar. Yeah, and what we really tried to do with our study was include a lot more people and make it more broader and more replicable, really, so that it applies to everyone.
And can you speak to that just a little bit? Because obviously, for many of us, we see lots of scientific papers. And my understanding in the scientific community, there's a bias toward that. Like when you find something, you want to publish a paper. But when you do all this work and you say, we didn't find anything,
yeah,
maybe it's not as exciting or sexy, but it sounds like it's incredibly important to do that type of research.
It is very, very important. And there is a movement at the moment towards more studies that replicate key findings. And Dorothy is a bit of a champion of it. So she's very involved in the UK in replicating studies, especially where there's in psychology. But I know from experience that when you're a geneticist, you want to find answers for people and you want to figure out what's going on. And it sometimes means that you keep going till you find an answer, and it's not necessarily the best way of doing things, but you are very driven to find answers for people.
Well, we really appreciate that, certainly for the XYY community, because there were studies done many, many years ago, which really gave a false impression of what individuals with XYY, what their lives would be like. So we really, especially this type of research.
Yeah, I think that's so it's still got implications now. And like, as I said about if you get a diagnosis here, they do tend to be like, done, problem solved, which I don't necessarily think it always is, especially with people with very profound difficulties, that it is worth keeping going and doing a thorough check of these things. And then it might provide answers for that person, but also for the family if they do have something that they are carrying within their family. It's important for everybody.
And somebody asked, so given, with that type of thing, so the individuals who had the difficulties with language development, who were in the other group, were all of them checked for sex chromosome trisomies?
Yeah, they were. So interestingly, we actually Diane did another paper where if you take a group of children with language disorders, you will find more than you'd expect of children with a trisomy. So it works in the opposite way around, that children with language disorders often are more likely than average to have a trisomy, but also people with trisomies are more likely to have a language disorder. So it sort of fits together quite well.
Okay, thank you. I'll find another. So would it have been possible to divide the group into a poor or average performing group to see whether there was a relation with CNV burden, as was found in the other study?
So that would be really interesting to do, but what would happen then is you would lose the power of the robustness of it. So it's very tempting to divide into different trisomy subgroups to see if there are differences between them. But every time you subdivide your groups, you lose the statistical power. So we were very careful not to do that. And it's a super interesting question. And I'm sure whoever does the study with 2,000 people in will be able to do that.
Is there going to be follow-up with any of these groups over time to see what interventions have happened and to see what will happen to these individuals?
I actually don't know that. So I think Dorothy Bishop is still quite involved, and she sends out a newsletter to all the families every year to keep them in the loop with what's happening with the research. I don't actually know the answer to that.
Okay. Do you think there could be any bias exists? So for example, where one provider might have said someone had a severe impairment versus someone saying not?
Yeah, and there's a whole host of different tests for each of those different conditions. And one clinician may say somebody is mild and somebody else might think they don't have a problem at all. So I'm not sure what it's like in the US, but in the UK, it depends on where you live, which hospital you go to, which person you go to sometimes. So absolutely, yeah.
And in the US, it also can quite often depend on if you have medical insurance, what access to care you have. And then the other question, I guess, would be is over time. So now, and I'm sure it's true everywhere, there are more genetic tests being done on individuals prenatally. And
Yeah.
watching more. Do you think that's going to change what we feel like is the overall, what we know about many of these conditions? So for example, when we're writing the CME course, because right now it would assume that people who are more severely affected, those are the ones that are getting diagnosed. As you said, they have a learning disability or some language disorders, versus
Yeah.
those that are not affected are probably under the radar.
Yeah. It's an interesting one. I actually don't know what would happen with companies like Ancestry and ones that you can as an adult get sequenced or get tested. I don't know that they would actually check unless you went through a hospital or the sort of formal system. I don't know that any of them would really check for something like a trisomy. Because that would be very complex ethically.
Right.
Yeah, but I imagine that people will eventually find out because they've done a test of that nature, and it's awfully complicated, and yeah.
And that's, and I know, speaking with some of the scientists, that's what they're saying. We just, a lot of things now is the group that we know about, and until you can't test everyone.
Yeah, but it also would have the advantage of being able to give much more accurate population occurrence predictions, which I think is a really important one, that it's poorly understood. And even though it's been known about for a really long time, I think it's actually quite a neglected area of research.
Absolutely. Did you see high verbal scale and low performance scale numbers amongst this data?
Not specifically. I didn't show the performance one. That was the dog photo. I don't actually have that. I'm not really sure. I'm not sure about that, I'm afraid.
Okay, someone else asked, were all the subjects non-mosaic?
Yes, we didn't include any mosaics because that would have potentially confounded it, because I guess you can be more or less mosaic. But that's another interesting area for future research.
And what about on individuals with XXYY, so somebody with 2 extra chromosomes, not just 1 extra?
So we only stuck to trisomies. We didn't look at any of the other subtypes. Again, they'd all make really, really interesting studies to do in the future.
Okay. So someone asked the question is how do you know if you see your physician, is the information about you going to these larger data banks? And I guess, and another question would be, I know with Access, we are looking into, one of our researchers is wanting to put together a patient registry with this information. So you can speak to that a little bit.
So if you go to your doctor and you get tested and you get diagnosed genetically, you don't appear on these databases that we refer to. So these are specifically for people who have sort of non-standard or unusual copy number changes, so that you probably wouldn't be reported in that way. But there is a lot of data sharing that happens between clinicians. So because there's a power in numbers, but they usually would ask your permission to do that, I think. I'm not like 100% sure on this, but I think that's usually how it is. In a research context, they will ask you if you're happy to be in a study. So you wouldn't get enlisted without your knowledge.
And do you do studies on adults?
No, this was just on children. So it really did sort of stem from the language disorder area of interest, and then went from there. So that's always mainly focused on children.
Okay. Well, thank you. Just want to really appreciate your being here, and thank you for your time. And thank you everyone for coming. We will be having this recording. We'll send it out to everyone who signed up, and we'll put it on our YouTube channel. So thank you very much for your time and for this.
Thank you.
All right. Thank you everybody for attending. Have a great rest of your day.